Hello, my name is Florian Boecker. I'm a postdoc at the Crop Bioinformatics group of the Institute of Crop Science and Resource Conservation at the University of Bonn in Germany. And I want to show you AHRD, our program for protein function transfer and evaluation. So our goals with AHRD is to do protein function prediction on a whole proteome-wide level. We need AHRD to be fast and easily scalable. We don't only want to annotate Go terms, but also descriptions. And both coverage and quality are important. For any prediction, it's always the question, how good is this prediction? So we want to do evaluation of protein functions as well. For gene ontology terms, we need to take the whole Go graph into account. And another problem with evaluation is, of course, always what do you use as ground truth? So AHRD uses sequence similarity for the function transfer, for the prediction. You can do that by running BLAST or Diamond, but with the sequence similarity search results, you can't always just take the top result. It's better to actually look at the whole result list and form a consensus from all database hits. And another thing with proteins from public databases is that some of the proteins in there have been annotated by other programs or even our program in the past by transferring annotations from other sources, and these indirect annotations are then of course not very desirable for us. So our solution to this problem is that we first filter out these kind of annotations by looking at the descriptions, and if they start with putative or uncharacterized, they're gone. In the second step, we ignore words from the descriptions that are very, very common to protein descriptions in general. We score each description by 3 factors: the alignment quality, the frequency of the words it contains in the whole search result, and AHRD can use multiple databases. We then can also distinguish between databases where the confidence in the descriptions is very high or low. For gene ontology terms, we also look at these 3 factors, but in addition, we can look at the evidence code of the annotation and use this as another avenue to try to get rid of annotations or avoid annotations that have been transferred from other proteins in the past. So this example protein is then annotated with this top scoring result, and we can now compare it to the ground truth to see how well this prediction did. And for the description, we matched 3 out of 4 words, but for the gene ontology terms, we only matched 1 out of 4 terms. But with gene ontology terms, you always have to keep the Go graph in mind. So if we look at a small excerpt of the molecular function Go graph, you see that the metal ion binding site term in the ground truth was actually indirectly annotated with our prediction of zinc ion binding. And similarly, the nucleic acid binding ground truth term was also reproduced by the prediction with the DNA binding term. It's just that the prediction was a little bit over-specific. So our solution to evaluate protein predictions incorporating the structure of the gene ontology graph is a semantic similarity Go score. To calculate it, we first need the information content of all gene ontology terms. We do this by looking at the annotation frequency of the terms in Swissprot, and then using this as an estimate for the probability to see those Go terms, and by taking the negative logarithm of that, you derive an information content. Now with the information content, you can look at pairs of gene ontology terms and determine the most informative common ancestors of both. And we use this to calculate the precision and recall, and from that our F-score. So the precision is calculated by taking the information content of all prediction terms and dividing it by the information content of the most informative common ancestors with the ground truth. And for the recall, we take the information content of the ground truth and divide it by the information content of the most informative common ancestors with the prediction. So this evaluation metric was also used in the artificial dilution series we did for our ground truth set. This method is pioneered by Plysin et al. From the Finnish group that is also behind Panzer, another prediction tool for proteins. And here our semantic similarity Go score is plotted against the signal level, and the rank correlation between the two is 0.969, which is in line with some of the top performing metrics in their paper. And we also looked at false positive sets, and from the score derived from this false positive set you can estimate a signal level, and with a signal level of 1.64 AHRD also fares fairly well. So our ground truth set is derived from SwissProt. First, we try to get rid of as much proteins as possible that probably have their annotation transferred from other proteins by only keeping proteins that have at least one experimentally verified Go annotation. And then we remove proteins that have putative or uncharacterized or other telltale signs in their descriptions. We then remove as much redundancy from the set by looking at the descriptions and removing all proteins that don't have at least one unique token in their description. And we also cluster the sequences and remove sequences that have more of a 40% sequence identity. So now I want to show you our results. We used 1,000 proteins and calculated the semantic similarity F-score for our annotations and the annotations of some of our competitors. And as you can see here, if you order these scores, you see that only AHRD and Best Bar SwissProt can annotate a significant number of proteins at a nearly perfect score. And AHRD also has a very good coverage. It's only bested by NetGo that has a coverage of 100%, but it does so by trading in a lot of the quality in the annotations. So descriptions were also annotated and compared to the descriptions of competitors. And you can see here that AHRD has the highest number of annotations that score with a 1, and also AHRD has the highest coverage. We always said that AHRD is easy to use, but now it's even easier. We implemented a SnakeMake workflow running AHRD. SnakeMake is a workflow management utility that also uses Conda to manage all dependencies. So the only thing the user has to do is to input peptide FASTA files. AHRD or the AHRD SnakeMake workflow then downloads the needed databases and does the sequence similarity searches, gets AHRD, builds it and runs it, and then spits out the annotations for your query FASTA files. And you can not only do that for one peptide FASTA file, but for dozens of eukaryotic proteomes at once. If you have the hardware for it, a cluster would be ideal, because AHRD SnakeMake is capable of deferring all these different tasks to different cluster nodes, and then you can rapidly re-annotate huge amount of big proteomes at once. And this is really important, because with sequence similarity, the one way you can actually increase the quality of your prediction is by having new information in the databases. So in order to benefit from new database releases, you have to re-annotate a proteome on a regular basis to be always up to date. And with the AHRD SnakeMake workflow, you can do that really easily. We also have a virtual poster with the same title and the same content. And you can find AHRD on GitHub and also the new AHRD SnakeMake workflow. Bye!